So let's take, because the beta cells are unique in that they are so metabolically active and they require these unique handling strategies to handle all of this load, it's important to see all of the different parts of the beta cell that are making ROS. So we've got four here. We've got the peroxisome in the top left. Peroxisomes are a organelle specifically designed in not just beta cells, in a lot of cells, to oxidize and neutralize free radicals, right? So it's taking things that could potentially be dangerous and makes them less dangerous. It's part of digestion, right? If you're digesting food, if your cell's using energy, it's kind of like a place where waste can go to be neutralized. Either way, it's a place where there's going to be a lot of ROS. Peroxisomes specifically are involved in beta oxidation of free fatty acids, or the fats that we're eating and the cell is using. So peroxisomes are creating that hydrogen peroxide. We mentioned before mitochondria, clearly very, very energy dense, therefore creating ROS. So what I want to pay attention to here is that this arrow here, this MNSOD, okay? So through the process of cellular respiration, there's a couple different ROS being made. Superoxide anion is one of those ROS. But like we mentioned before, hydrogen peroxide is the preferred ROS because of how stable it is. So our body has an enzyme in place called manganese superoxide dismutase. And so this SOD, this superoxide dismutase, is something that takes that superoxide anion and turns it into hydrogen peroxide. Okay, so it's making a less stable ROS into something more stable that it is able to neutralize via this GPX enzyme, turning it into water and oxygen, which is something that is not toxic to the cell. Now, the endoplasmic reticulum is also creating its own free radicals. I mentioned before that the endoplasmic reticulum in the beta cell is larger because of that high hydrogen peroxide content, because of how active these mitochondria are. So they themselves are metabolizing hydrogen peroxide, as well as in the cytoplasm, the cytosol itself, just within the cell without actually being inside an organelle. Any superoxide anions that are byproducts of other cellular events happening in the cytoplasm are reacting with a different SOD. Notice how superoxide dismutase, that's something that's taking that superoxide, turning it into hydrogen peroxide, except this one is a copper, that's what Cu stands for, and zinc SOD. So just between the mitochondria and the cytosol here, notice how available and, well, how apparent trace minerals are, right? Manganese, copper, and zinc. Those are all things that you recognize from, nutrient labels and nutrition labels, right? So our body is using those trace minerals that we ingest from food and using it to neutralize oxide, the superoxide anions, into hydrogen peroxide. Now, the real important part about this and where toxicity comes in is the fact that when that superoxide anion is converted into hydrogen peroxide, this hydrogen peroxide is liable to react if there is free iron present via the Fenton reaction, which we have talked about extensively on this podcast, right? Hydrogen peroxide and iron is like a match to dynamite. It is something that makes it so much more toxic, right? So this OH dot here that you see in the mitochondria as well as in the cytosol, that is the hydroxyl radical. That is the Tasmanian devil version of ROS. It is so destructive. It is it just blows up in place. Your body cannot react quick enough to neutralize the damage, which is extensive, that hydroxyl radical can do. And because the mitochondria is a place where a lot of energy, a lot of waste, and a lot of iron is coexisting, it makes for some precarious situations, especially if you do not have the correct systems in place to neutralize those very toxic components, right? So the mitochondria, high energy, or beta cells in general, not just mitochondria, beta cells in general, high energy cell, creating a lot of waste. That high energy requires a lot of iron to convert to make this energy. A lot of that waste means a lot of ROS. So you have a lot of iron and you have a lot of ROS. Can we see the potential for danger here? Can we see the potential for disaster when you have this iron and this ROS, when we know that those two are the ingredients for creating the hydroxyl radical? And so here we are. Check episode 11 and 12. I've gone into this more extensively before, but the Fenton reaction is essentially where free iron is reacting with hydrogen peroxide here and creating the hydroxyl radical, multiple hydroxyl radicals, 2 to be specific, but it's essentially compounding what was once a stable ROS that could be readily removed from the cell and has turned it into 2 highly destructive, highly reactive molecules. Bad news for a cell. Bad news. Okay. So plenty of documentation on how destructive hydroxyl radicals are. Plenty of research going into the Fenton reaction. It's not just a bodily event. It's used commercially, industrially. It is an explosive type of reaction, and it has a very well documented bodily harm. Bodily harm like DNA and RNA deconstruction, mutations. It breaks up and destabilizes cell membranes. It tears apart proteins, enzymes, and it is, I'm trying to think of the right word for it. Nondescript. It is indiscriminate. Just indiscriminate destruction. Okay? Anything in its place, it's getting rid of. So when you think of ways that this could materialize in the state of diabetes, we have our beta cells with a lot of iron, a lot of ROS, and when we're diagnosed with diabetes, we're kind of like entering the crime scene and everything's kind of destroyed. Meanwhile, no one is looking to check for the hydroxyl radicals' fingerprints when logic would tell you that they have been there.